All right. Hi, everyone. I know people are still filtering in a little bit. Sorry that we're running a few minutes late here, but welcome to our KCART conference or our KCART seminar for at least for this week. So just wanted to give a couple of housekeeping notes. So if you've been here before, you know the deal, but we do ask that everyone remain muted during our presentation today, but you are welcome and encouraged to leave your videos on if you want to. If you have questions for our presenter today, we do ask that you can either send them in the chat and they'll actually go to me and then I can pose them to our speaker at the end of the talk, or you can wait till the end and unmute yourself and then ask your questions directly. Without further ado then, I'm going to pass it off to Dr. Mosconi, who's going to be introducing our speaker.
Great, thank you, Shannon. I'm delighted to get to introduce Dr. Cascio for today's research seminar, but before I jump into the introduction, just a couple of quick bookkeeping or scheduling notes to share. So we've had a bit of a gap between research seminars, so it's good to see people back. The upcoming schedule, there are two events scheduled in the coming weeks. 2 weeks from now, from today, Nicole Zieber will be doing her presentation on early behavioral infant detection of emotion from facial expressions. So in 2 weeks, this same time, we'll get to see Dr. Zieber's talk. And then next week, I did my best to make this a virtual background in Zoom, but it's just not great unless I duck. But our Autism Across the Lifespan conference is next week. And for those who have not registered, it is not too late to sign up. We'll save you a seat. It is a virtual meeting, so that's just a metaphor. We look forward to people being there. Our keynote speakers both actually happen to be from Vanderbilt, where Dr. Cascio is coming from as well. We keep going back to that well, but it's because there are good people and good scientists there. But Doctors Jim Bodfish as well as Julie Lowndes-Taylor will be our keynote speakers. Dr. Taylor will be speaking in the AM, and Dr. Bodfish will be speaking during the lunch hour as our keynotes. But a number of KU Med Center, Children's Mercy Hospital, as well as other investigators and partners within our region will also be speaking throughout the day. So it is a full itinerary and we hope to see you there. So I am very excited to be able to welcome Dr. Cascio, Carissa, to our research seminar today. Dr. Cascio is coming from Vanderbilt, as I mentioned. She's an associate professor there and actually has been there at Vanderbilt since 2007. She originally did her undergraduate work at Baylor University in neuroscience, then completed her PhD at Emory University. I got to meet Carissa for the first time back in about 2006, 2007. Yes, we're old, Carissa. When she was doing her postdoctoral training there, in neurodevelopmental disorders. And it's just been wonderful to communicate and stay in touch with her and even do a little bit of work with her over the years since then. I've tracked her work closely personally just because I've always been so impressed with the rigor of the science, really. And Carissa was one of the people that was really quite important to advancing our understanding as well as our emphasis on sensory issues in autism. When Carissa was studying sensory issues in autism, they weren't part of the DSM diagnostic criteria yet. And I think her work was important to making that advancement and changing how we think about autism in many ways. Her research focuses on the neural basis of sensation and perception. She's been funded by NIH for a long time, really, at each stage of her career, starting from a K Award to the present, where she is, I think, in year 7 or 8 of an R01 award and its renewal. And those studies have focused on attention and and sensory perception in autism spectrum disorders. So I'm very excited to welcome Carissa to our research seminar. Looking forward to your talk, Carissa.
Thank you, Matt. Thanks so much. I appreciate that's a very kind introduction. Despite the fact that you called me old, I'm going to overlook that. But it is really wonderful to be here virtually with you guys and to see all the familiar faces of scientists that I really respect and value, and people that were trainees at Vanderbilt and are now part of your center, and old friends from postdoc days. So thank you so much for the invitation. It's wonderful to be able to share some of the stuff that we're working on in the lab, and to hopefully get your feedback on it, because it's all a work in progress. So I'm looking forward to that. Like a lot of people, I've taken to starting with the acknowledgement slide because I often find myself running out of time at the end. So I'm going to take a moment to acknowledge my lab, the Laboratory of Affective Sensory Research, or LASER, as we've now branded ourselves. Most of the work I'll talk about today was done by these lovely people that you see here, many of them whom have now moved on to other posts. I'll highlight each of them throughout, though, when I'm talking about the relevant work. And then my collaborators at Vanderbilt. So at the Imaging Institute, our main collaborator is Baxter Rogers. And then Mark Wallace, and a graduate student, at the time, a graduate student, John Paul Noel, working with Mark, and who is now at NYU in a postdoc position. So, and obviously our funding and our research participants are always sort of among our major people to thank. So as Matt highlighted, we have a lot of common ground in terms of a focus on sensory and sensory motor differences in autism. So, you know, to some extent, this is sort of well-worn territory for this group. But I always like to highlight the fact that a lot of what we point to when we think about the fact that sensory differences are prominent in autism are accounts from people with autism themselves. So their own accounts tell us that their sensory experiences and their emotional reactions to those experiences are different from the neurotypical population. And often this is described in terms of really positive emotion or really negative emotion. So in the case of Temple Grandin's example of the school bell sounding like a dentist drill going through her ears, you could see how that would be unpleasant to the point of really interfering with a lot of other things. But sometimes these differences are described as pleasant or rewarding, and maybe can give us some insight into repetitive behaviors, as in this description from Naoki Higashida from The Reason I Jump. When I'm jumping, I can feel my body parts really well, my bounding legs and my clapping hand. That makes me feel so, so good. So, you know, on the one hand, negative experiences, and on the other hand, positive experiences can give us a window maybe into some of the more basic mechanisms that lead to the things that we see in terms of social withdrawal or repetitive behaviors, or at least that's the hope. And this idea is not new, you know, even though we, you know, we've struggled maybe to draw attention to it. Back in the 70s, Orniz described this unusual reliance on proprioceptive and tactile cues in children with autism in learning contexts, and actually made the suggestion that repetitive behaviors were adaptive behaviors that helped them to make sense of both internal and external sensory information. But as Matt kind of alluded to, for a long time, this perspective really took a back seat in the research world because we were much more focused on, you know, what we think of as the core features of the disorder, the social communication skills, and how, you know, explosively those are developing during early childhood, and how we really wanted to understand that trajectory to understand what was happening differently in autism, you know, which was all really fruitful areas to research, but it really kind of put, you know, these basic sensory and motor features in the backseat for a little while. But with the advent of you know, during all that time, me and many other researchers were continuing to try to understand the sensory aspects of autism. And with the DSM-5 revision, I think the attention to those features has really increased. But we're still really trying to work out how best to study this. So by definition, studying sensory processing requires us to understand a process that is largely an internal process. And so an example that I like to use is these two sisters who have a YouTube channel where they taste test different foods and they give their impressions to other kids. And so in this frame, they've both just tasted a lime. And looking at them, we might say that their sensory experiences were really different, right? And maybe that's true, but presumably they both have taste receptors in their tongues that responded to the sudden drop in pH and relayed information along the cranial nerves to the brainstem and the thalamus, and then finally up to the anterior insula, where primary taste cortex is. So, would we expect there to be differences along this pathway, or did those differences emerge later when higher order cortex interpreted the meaning of the sour taste or compared it to previous experiences or integrated that input with information about who was watching or what their expectations were? So it's clear from this to me that especially when measuring behavior, it becomes really difficult to separate what is sensory from what is emotional or attention or personality or all sorts of other factors. So we have a lot of tools that try to help us with this challenge, and they fit together having different strengths and limitations. So observational measures and caregiver report are really strong on ecological validity, but they have this limitation that I just described. We're using a third party to make decisions based on really an external behavioral response and making assumptions that those map directly onto sensory experience, which they may or may not. For individuals who are able to self-report, questionnaires unfortunately often do a pretty poor job of separating these factors as well. Even though the person is reporting on their own experience, there's often sort of conflation in the items themselves among these different aspects. And then psychophysics is a technique that we use in experimental settings where we have controlled presentation of precise sensory stimuli followed by a judgment about them. So for example, was the first tone higher or lower in frequency than the second, or press a button when you feel a vibration on your skin. And so these methods are the absolute worst on ecological validity. Nowhere in real life do you have a single vibration happening on your skin with nothing else going on. But to some extent, they allow us to separate out what's sensory from what's other things. But they also have some assumptions that may not be valid when we're talking about clinical populations. So they assume consistent attention within individuals and across individuals, and consistent abilities in motor control across participants, which we know is probably not a valid assumption. So what we tend to try to do is combine all these things and try to look at it along with neural measures to try to get a clear picture of what's happening in the sensory worlds of people with autism. So in addition to the challenges looking at behavior, our understanding of how the brain is processing this information is also changing. So when I was first learning about sensory processing in the brain as an undergraduate student, we might have described it something like this. There's a stimulus in the environment, and that creates a sensation that rises to the level of primary sensory cortex, and then association cortex gives you a perception and interpretation and evaluation, and eventually that sort of sends something to motor cortex that gives us a reaction. And while none of that is necessarily untrue, our current thinking of how the brain is treating sensory information is a little bit more complex. So incoming sensory input arrives at these waypoints and is cross-checked against predictions made based on previous sensory experience. And this sort of results in a prediction error, in this case sort of indicated by the letter E. And this sort of happens throughout, one instance of sensory processing and then over time, so that based on our previous sensory experiences, we're telling lower order regions of cortex what to expect, and then looking at how well the actual input matches that expectation. So this gives us a much more complicated and less linear process of brain processing, but it's also one that's more flexible and plastic. And so our way of thinking about how the brain is dealing with sensory information has changed. over time. Our thinking about what's included even under the term sensory has expanded and changed over time as well. So many of the researchers in this group are already very familiar with this concept, but you know, a lot of times sensory is kind of considered to be, well, the world out there. And it's actually more than that. There's a rich sensory tapestry from skin and muscles and tendons and viscera. Much of Matt's work on the motor system has highlighted this. And these are also of interest in autism. And in my lab, we kind of started somewhere in the middle of this distribution. So I kind of order this in terms of where the stimulus energy could be coming from. You know, and for vision, that can be, you know, millions of miles away, and all the way over to vestibular and interoception and proprioception, where the sensory cues are actually coming from within the body. So in my lab, we kind of started with this idea of touch and the skin, have kind of journeyed inward to the interior of the body by expanding our focus to interoception, which is mostly what I'll talk about for the rest of the time today. So what is interoception? Well, interoception is the perception of sensory signals from the viscera, including the feelings from heart, lungs, bladder, the digestive system. And it's an unusually constant form of sensory input that's often really sort of filtered out of our conscious awareness, kind of a quiet drumbeat that we really habituate to, but occasionally is kind of punctuated by perturbations where these things rise to the level of our conscious awareness. And the examples of that include when our heart starts to race or we get butterflies in our stomach. And a lot of these perturbations where our physiology starts to capture our sensory attention are linked to emotional experiences. And so to illustrate this further, there's a whole lot of our language that we use to describe our own emotional experiences that really reference interoception. And I've put some of my favorite examples up here. Physiological changes are often signaling events that are really meaningful to us in a very fundamental biological sense in terms of our safety or well-being. And that's really tightly linked with our emotional responses. So about 5 years ago or so, researchers started to wonder if autistic people experience interoception differently, as they seem to do for exteroceptive sensory signals. And there was definitely more than one way to form hypotheses about this question. So because we just talked about how sensing these internal cues is related to emotional experience, and people with autism sometimes have trouble interpreting their emotions, you might predict poorer interoception in people on the spectrum. But if you think of interoceptive and exteroceptive signals as competing with one another for attention, you might expect that somebody who tunes out or is overwhelmed by external input might be actually relatively more sensitive to interoceptive signals. And we've certainly seen that kind of relationship in other paradigms where proprioceptive and visual inputs are paired. Or it could be that interoception is not affected in autism either way. But just as for exteroceptive modalities, we have to be clear about how we're defining and measuring this construct. So Sarah Garfinkel, who's at the University College of London, proposed the following model, which has come to be pretty widely used and defines interoception based on 3 dimensions: accuracy, sensibility, and awareness. So in this model, interoceptive accuracy is measured with the kind of experimental psychophysical paradigms that I referenced earlier, measuring most often heartbeats and asking people to count or track them, versus interoceptive sensibility, which is measured by self-report of how often one notices various interoceptive signals. And finally, interoceptive awareness is a kind of a metacognitive measure in which your actual performance on an accuracy task is measured against your confidence in that performance. So for each trial of a heartbeat detection test, you might be asked how confident you were in how well you did. And if there's a large discrepancy between how accurate I am and how confident I am, then I'm said to have poor interoceptive awareness. So this is kind of the model that most of us are working under. And as I mentioned, most of the experimental work on interoception is done on heart signals, and this is really just a practicality of that they're the easiest to measure relative to other organs. And so a short overview of how the heart functions will help to understand the methods a little bit. So this is probably a review from high school biology class for most of us, but the atria of the heart are where the blood is received up here at the top, and the ventricles pump it out to the body and to the lungs. So the right atrium receives oxygen poor blood from the body and pumps it into the right ventricle, where it goes to, where it's pumped out of the ventricle to the lungs for oxygenation. And then oxygenated blood from the lungs is coming in through the right ventricle, goes into the left ventricle to be pumped to the rest of the body through the aorta. So that is kind of what the cycle of the heart looks like. And this is sort of that same information just mapped to the different parts of the EKG. We can measure the electrical impulse that creates these contractions as it travels through the heart. So this little tiny bump represents the contraction of both atria, and the sort of signal travels from this sinoatrial node down towards the ventricle. And you can see these little sort of waypoints represented with these different peaks. But what we're really most interested in is the contraction of the ventricles that pumps blood out of the heart. And this phase is also called systole, and is represented by this large peak called the R wave. And so this kind of shows the same thing with the whole cardiac cycle as represented by the EKG. The peak of the R wave is what anchors what we think is the most significant source of sensory input as blood is pushed into the aorta. So where are the sensory signals coming from? Well, there are mechanical receptors within the ventricles that fire as it actually contracts. But we really think that the bulk of the sensory input to the brain is coming from baroreceptors or stretch receptors in the aorta and the carotid artery. And that pressure, those signal pressure changes with each bolus of blood that is ejected from the heart. On average, this pressure change that activates the baroreceptors, we think the signal reaches the brain on average around 250 milliseconds after the R wave. So that's a little overview of the source of the sensory signals that we're trying to measure. There's several experimental paradigms that have been designed to get people to pay attention to their heartbeats and understand to understand how they're perceiving them. The simplest one is what's known as the Chandri task, or the heartbeat counting task. In this task, a continuous EKG is recorded over an interval of several seconds. So it might be 10, 20, 30. Usually there's several different intervals in a single experimental session. And it's just a very simple concept. You ask people to concentrate and attend to their heartbeats without feeling their pulse over these intervals. And then you just calculate an accuracy metric by comparing the reported number of beats to the actual beats that you get from the EKG. So it's stupid simple, and it's therefore really widely used, but it has some limitations that we'll discuss in a bit. But this was the task that and I will say that all of the interoceptive literature in autism so far has used this task. And Garfinkel herself was sort of the first person to look at this in autism. So she had 20 adults with autism perform this task and found lower interoceptive accuracy overall in their autistic group relative to a comparison group. So their autistic group here is in this teal color. Their comparison group is in the peach. And so the heartbeat tracking accuracy score is much lower there. In contrast, they found greater interoceptive sensibility in their autism group. So this was measured with the body perception questionnaire, which again asks questions like, how often do you notice that your mouth is feeling dry? How often do you notice that your heart is racing? All these kinds of questions about, in your daily life, how much do these signals grab your attention? And so people with autism reported that those signals grab their attention a lot more than the comparison group, despite being less accurate at perceiving them. Garfinkel and her colleagues also reported that across groups, what they defined as trait prediction error, or the magnitude of the difference between the previous two measures, was significantly correlated with anxiety, suggesting that the disconnect between the attention toward and the accuracy of perceiving these internal cues contributes to anxiety. Around the same time, our lab was using the same task in a sample of children with autism. And this work was largely done by Kim Schauder, who was a research assistant in the lab at the time. Kim actually did not find a group difference in interoceptive accuracy in a sample of children that was similar in size and to that in the Garfinkel paper. In fact, when she looked at the longest interval we collected, which is 100 seconds, and that's a pretty long time to stay quiet and attend to a person's own heartbeat, it seemed that our autism group actually maintained performance where our typically developing comparison groups suffered. So over that long period of time, it's really common to see interoceptive accuracy drop off because it's such a long time. But we in this sample didn't see that. We saw sort of a kind of a maintenance of their performance relative to their overall performance and relative to the shortest interval. And so this made us wonder if maybe kids with autism were better at tuning out external distractors while focusing on their heartbeats, or had some other kind of advantage for internal sensory cues. So we did a follow-up experiment where we explicitly tested that and presented a vibrotactile distractor while people were doing the heartbeat counting task. And so unfortunately in this test, we did not sort of support that hypothesis. So there was no evidence that people with autism were better able to tune out these exteroceptive distractors and maintain focus. So whether we used a weak distractor with a low vibrotactile amplitude or a stronger one with a higher amplitude, performance decreased slightly relative to the no distractor condition for both groups. And that didn't necessarily vary by the intensity of the distractor or age in a significant way. So while we were exploring this sort of attention thing, a few other studies came out, unfortunately with equally inconclusive results in terms of a primary interoceptive deficit in autism. So overall, the weight of the evidence doesn't support this middle idea that there might be increased attention to or accuracy for internal sensory signals. But there's kind of equal weight on the sort of just null result and deficit side. So studies in both children and adults vary in which they find no effect or decreased accuracy. So as I mentioned, the task used in all of these studies up to this point, the Chandri task, has some limitations. So what we have working for it is its simplicity. And that's led to widespread use, which kind of conveys an additional advantage, that it makes it easy to compare findings across multiple studies. And we also liked it for what we thought of as its interoceptive purity. So you're just asking people to focus on heartbeats and report number of heartbeats, and there's really no other thing that they need to pay attention to, which is in contrast to all the other alternatives to it that we'll talk about in a second. Working against it, though, are several factors that might influence how people count the beats. So people may already have an idea about how fast their heart should be beating. If you get that information from the doctor or from your Fitbit or your Apple Watch, you probably have some sense of what your heartbeat should be. And so you may guess based on that estimate when you're doing this really hard perceptual task, rather than trying to perform the task as intended. We try to address this by debriefing and kind of excluding people who report that kind of a strategy, but that's certainly a limitation. Additionally, most people, well, I would say that the working memory load is probably not negligible as well. So we're asking people to sort of count and hold in their memory over, intervals of several seconds. And so that's another potential limitation. But then one that we're really sort of focused on, and which we think might be confounds that directly impact people with autism, is that there's a directional asymmetry to this. So most people can't feel every beat. And so when people make errors, it's usually in the direction of underestimating their number of heartbeats. And that favors people with lower heart rates. And on average, people with autism tend to have higher heart rates. Similarly, it's a perceptual detection paradigm with a kind of weak signal that's close to threshold. And so people consistently, it favors people with a liberal response bias. So in other words, if a sensory stimulus is near threshold and thus uncertain, people vary in how likely they are to say, yes, I detected it, or no, I didn't. And so we've found in other tasks that people with autism tend to have a more conservative response bias. And so that could be another reason in some studies they appear to perform more poorly on this. So as I mentioned, there's some alternatives to the Chandra task, and all of these are variations on a theme of trying to detect your heartbeats and compare them to an extraceptive periodic stimulus. So a series of tones or flashes. We're currently running several of these alternatives in the lab, but before we even really started to think about that, a grad student in Mark Wallace's lab asked, and this is JP Noel, if we could do a similar thing by trying to measure a cardiovisual binding window. So the Wallace lab focuses on temporal multisensory integration, which is a phenomenon in which the brain binds stimuli from two sensory modalities and perceives them as arising from a single cause. So this is what is behind you being able to see my mouth moving and hear my voice and not really think of those two things as two separate things. And Mark and his group have established that people on the spectrum have unusually wide windows of time in which they bind sights and sounds. So JP was curious if we could measure a temporal binding window for interoceptive and exteroceptive stimuli. When he first asked about this, I was kind of reluctant. I was like, I don't really think we should do that. It's really clear why there would be an advantage to binding exteroceptive stimuli like voices and mouths, or even binding like proprioceptive to exteroceptive stimuli, like for controlled movements that are guided by vision. But what on earth would be the advantage to binding cardiac and exteroceptive stimuli? I just don't see it. Well, he convinced me and we tried it anyway. And so what he did was present the visual stimuli at random intervals while we measured the heartbeats. And then offline, we kind of calculated the stimulus onset asynchrony, or the SOA, which was sort of the temporal discrepancy for each flash relative to the closest R wave in the EKG. And we asked participants to judge on each trial whether the flash was coincident with a heartbeat or not. And so for audiovisual stimuli, this is often what we might see. The offset between the two, as the offset between the two stimuli increases, the probability gets lower that they'll be judged as simultaneous. So if people do this task perfectly, you see a big, strong peak at 0 milliseconds and a sharp drop off as you get further away on either side. So the less spread around that peak at 0, the better they are at the task. You can see that this curve for audiovisual is wider at the tails and shorter at the top for autism, which suggests that they both fail to perceive truly synchronous events as synchronous as often as controls, and that they're also more likely than controls to call very asynchronous events synchronous. So what we see in the cardiovisual experiment is sort of an exaggerated thing. So you'll first note that this task is much harder. So the cardiovisual binding task is harder. There's not as sharp of a peak here, even for the typically developing group. But the differential between the TD and ASD performance is much more dramatic. So even though by all of our previous metrics, there's not really a clear primary deficit in perceiving interoceptive signals alone, this was convincing to us that people with autism actually really have trouble integrating those interoceptive cues with an external signal. So I went back to my question to JP and asked, well, why would this be necessary or important? And why would people with autism be so much worse at it? As we thought about it, one of the other students in the lab, Elisa Zoltowski, who's really interested in co-occurring anxiety in autism, suggested that comparing physiologic cues to the external environment might be really important for interpreting threats. So for example, if you feel your heart start to race and you look around and you see treadmills and hand weights and elliptical machines, associative learning is telling you, oh, it's okay, I'm at the gym, I'm running, and that's why my heart is racing. But if instead you look around and you see a microphone and hundreds of unfamiliar faces, you may conclude that your racing heart is signaling that you're afraid. They're actually not okay because you're terrified of public speaking. So an inability to integrate those internal and external cues could contribute to heightened anxiety or difficulty in interpreting one's emotions. And this is consistent with the data that we saw from Sarah Garfinkel's work. So as an aside, there's very new research that suggests that we actually have the ability to perceive other people's physiological signals as well. And so this was just something that I found super interesting that I wanted to throw in there. This study is from James Kilner's lab, also at UCL, that found that people perform above chance in matching a video of a person to the audio of their heartbeat. And determined that the way that they were doing this were micro variations in the color of the face that co-vary with the cardiac cycle. So here is sort of the peak of the R wave, and they measured sort of 400 milliseconds before and after that. And you can see that this is aggregated data across all of their people, but you can see that there's these changes that are, probably close to imperceptible on an individual basis. But that information is being perceived somehow, and people are able to use this information to be able to identify a speaker in a video or a face in a video with their heartbeat. So even though so far the bulk of the experimental evidence that we have doesn't really suggest a clear difference in heartbeat counting ability in and of itself in autism, we wondered if, knowing again the limitations of just the task and relying on behavior, if we might see a difference at the level of the brain's response to heartbeat counting. So we ventured out to do an FMRI study, and our primary region of interest is the posterior insula, which is designated as primary interoceptive cortex. So interoceptive input travels from the brainstem, the baroreceptors input to the brainstem, to the ventromedial nucleus of the thalamus, and then the posterior insula is where that thalamic nucleus projects. The insula is a highly gyrified region that's located sort of deep buried within the lateral sulcus, and it's been implicated in a number of functions ranging from sensory to cognitive. And in most mammals, including humans, it's notable for its varied cytoarchitecture. It's typically divided into 3 regions: agranular, dysgranular, and granular, which refer to the absence, low concentration, and high concentration of granule cells in layer 4, respectively. The human insula has a granularity gradient such that the anterior ventral insula is agranular, while the dorsal posterior primary interoceptive region is granular. And we think that those granular cells have a role in making computations based on sensory prediction errors. So the hypothesized function is that the granular cells amplify and distribute the thalamic information throughout the layers of the cortex, and then the agranular cortex, which is more visceromotor, sends predictions back to those granule cells based on previous sensory experiences, allowing the system to function sort of like a sensory motor loop to control visceral function. So to understand how this region is responding, when we ask people to attend to their heartbeats, we asked a group of people, including children and adults with and without autism, to perform the Chandri heartbeat counting task in the scanner. And here you can see it was a reasonably large sample for an FMRI study that was matched on age and sex. So we had 46 people with autism and 54 people with typical development. And we asked them to count their heartbeats for 30-second blocks or to count a visual stimulus as a control presented at a very low luminance to try to make it closer in difficulty to the heartbeat counting task. We pseudo-randomized these blocks with 15 seconds of rest between them. And we obviously were focused on the insula, and we used cytoarchitectonic maps to draw regions of interest where we were able to delineate the more anterior part of the insula, which we think is more visceromotor and connected to more cognitive and emotional regions, from the posterior part of the insula, which is the primary interoceptive cortex and more sensory. So when participants, oops, sorry, I didn't want that to be, when participants performed the task in the scanner, we were able to review their accuracy in a different position. So they were lying supine in the scanner as opposed to seated, like they were for previous instances of the task. Again, confirming what we had seen before, we found no differences in their accuracy of heartbeat counting for either children or adults with autism. I think, sorry, that was children and adults, and this is overall. Okay, so when we compared counting heartbeats to counting the visual stimuli, we saw that both groups recruited insula and anterior cingulate cortex, and there were no significant differences between the groups. So you can see that both groups have a robust activation in the insular cortex throughout the insula, sort of anterior and posterior, and in the anterior cingulate, which is highly functionally connected to the anterior portion of the insula. So it was encouraging to see responses where we expected to in this contrast, and we subjected the results to a non-parametric permutation test as a confirmatory step. And so this sort of further showed us that we have activation in the insula, response in the insula to this task relative to our visual counting control task, as we would expect. And again, no real differences in the extent to which that is true for people with autism versus people without. Our sample had a really wide age range, and so we wanted to know how age affected the bold response throughout the insula. So to investigate the associations between age and percent signal change in each of these divisions, we explored linear and nonlinear models, given previous work suggesting that both linear and nonlinear developmental effects are present in the brain. And so here you can see the kind of best fitting models for both regions, and they collectively suggest that the pattern looks pretty similar throughout the insula for both posterior and anterior and middle insula, and that there's this curvilinear relationship with age such that the response of the insula peaks when doing this task sort of in early adulthood and kind of the early 20s. And so this kind of coincides with evidence that autonomic function, so measured fluctuations between heart rate and systolic pressure, also seems to peak physiologically in this time period. So finally, given what we were hypothesizing about how interoception might relate to emotion interpretation or other relevant clinical features of autism, we explored the relation between the SRS2 and response to the posterior insula. So again, we had a wide age range. So we had caregiver reports for children, and we had self-reports for adults. And you can see that we saw very different things here. For adults who self-reported symptoms, there was a positive relationship between higher autism traits and more response in the posterior insula when counting their heartbeats. But the opposite was true for adults without autism. This relationship wasn't present in children. So, obviously for children, we didn't have self-report, we had parent report. So there could be several explanations for these disparities, but it was interesting to us that during the age span, when this response peaks in this kind of early adulthood, late adolescent period, people with autism who have the most robust response are describing themselves as most autistic, while the opposite was true of adults without autism. So this could suggest a fundamental difference in the functional nature of the of interoceptive attention. So there's a researcher who's working with Jamie McPartland at Yale named Dominic Trevison, and he's recently pointed out that heightened interoception can be adaptive, as it is in, say, meditation, but it can also be maladaptive, as it is in somatic disorders. And self-report questionnaires of interoceptive attention and sensibility are really different in terms of what, which of those two ways of looking at the construct that they tap. And so he's also showed evidence that reported differences in interoceptive sensibility in autism are limited to more maladaptive forms of interoception as indexed by self-report. So that dichotomy could be also really instructive to guide future work. So to summarize, using the heartbeat counting task, we don't really see a primary deficit in autism at either the behavioral or the neural levels, although there's probably some effect for interoceptive sensibility, particularly for maladaptive interoceptive sensibility. And it looks like there is an effect for interoceptive, exteroceptive integration. In our FMRI study, we found that age predicted the bold response across the insula in a nonlinear fashion, and that response in the right posterior insula was associated with self-reported autism traits. And future directions, we're currently continuing to explore the interoceptive, exteroceptive integration more thoroughly, using tasks that were really explicitly designed to separate these things out, and looking at that with clinical measures of co-occurring anxiety. So that is all I have, and I'm happy to take questions in whatever format people want to ask them. Thank you so much for your attention.
Thanks so much, Carissa. It's really interesting talk. I'll go ahead and just open up to the audience. If anybody wants to ask a question, please feel free to unmute yourself and ask Dr. Cascio directly. Shannon, are there any questions in the chat box? If not, I'm going to jump in.
No, I don't have any.
All right. So, Carissa, I actually, some of the early studies that you highlighted were particularly intriguing to me because, I mean, this is a relatively novel, I guess, direction within the field. The past 5 years, there haven't been a lot of people looking at interoception within autism, and then you had this finding, I think, from the Garfinkel studies showing a mismatch between the accuracy and the sensibility that seems to relate to anxiety. So I'm trying to understand sort of what that association may reflect. In other words, whether there may be like a true hypervigilance that drives, or hypersensibility that drives anxiety, or whether there is some other mechanism causing anxiety that then sort of has this feedback that that increases, I guess, sensibility but not accuracy to cardiac rate, to these interoceptive processes. So how are you conceptualizing that relationship?
Yeah, it's a really good question. So I think my default is to think of sensory phenomena as more being drivers of more complex phenomena. And so I think my assumption, my working model is that this attention to, and in the case of this, of her data on interoceptive sensibility, the body perception questionnaire is very much a measure of what you would call maladaptive sensibility. So, you know, things like, you know, I am really tuned in to, you know, when my stomach feels upset, or, you know, when my heart races, and, you know, nothing that is sort of like, oh, I'm able to focus on my breath and, you know, be one with the universe. And there are definitely interoceptive questionnaires that are more along those lines. So, you know, my thought is that this, you know, this kind of focus on that, and then perhaps the discrepancy between that focus and how accurate they are at identifying those cues is sort of contributing to anxiety rather than the other way around. But I think it's an empirical question for sure.
Yeah. I guess the other question that sort of piggybacks on that too is whether there is some level of variation within autism that that may relate to cognitive abilities or to IDD within your sample, because you do see such spread there, and you do see variable levels and manifestations of anxiety, you know, across individuals with autism that can co-vary with IDD. So I don't know if you've looked at that, and certainly, of course, that's always a challenge within the imaging studies because of the sampling population. Yeah.
So we unfortunately have not had the opportunity to look at that, but I am hopeful that we may get the chance to do that. So these kinds of the heartbeat counting task and that kind of thing, imaging is difficult for people with IDD. But one of the things that we're doing in the new study, the R01 renewal, is we're doing what's called a heartbeat evoked potential. So I didn't talk about this because we don't have data on it yet, but you can measure the brain's response to individual heartbeats by aligning the EKG and the EEG signal. And it's pretty cool. So we're getting that now. And that's absolutely something that just happens in the background without any sort of requirement for a task or anything. So we're hoping for at least that part of our study to expand our inclusion and be able to look at some people with lower cognitive abilities.
Right.
I have a question, basic question. Hi, this is Zore. Great presentation. And my background is genetics, so I apologize. This topic is above my understanding, but fantastic work. I wonder if there has been any differences between gender and the kind of assessment that you are doing in your projects. And you mentioned about anxiety, and I was wondering if sensory issues at this granular level has been shown to have any association with other comorbidities, such as sleep problems or GI issues, which you would think there might be some connection with sensory problems. Thank you.
Yeah. That's a great question. So in terms of sex differences, unfortunately, I don't have too much data to address that with at this point. So we've certainly looked, but I think we're underpowered with the sample sizes we have to really say anything meaningful about differences by gender or group by gender interactions. But it's certainly something that I hope that we will be better equipped to address in the future. But I absolutely think that there's a strong possibility that some of this is related to some of the co-occurring conditions that you mentioned. So sleep problems, GI problems, and that's certainly a direction that we'd really like to go in kind of future iterations, is to try to determine to what extent this kind of difference in interoceptive sensibility or perception might be influencing those things.
Great, thanks.
Hey, Carissa.
Hey, Brian.
Nice to see you again. So, I'm always interested in sort of the clinical aspects of this. And so it makes sense to me, and sort of as a hypothesis, that some of this discrepancy, if you will, between sort of internal processing or interoception and sort of how we're thinking about whether that lines up with the external input, with the processing of the environment, may help understand anxiety or sort of threat assessment, if you will. But do you think it also helps us understand in any way sort of the more traditional sensory sensitivities we think of within the context of autism, like their hyper-responsiveness or either under-responsiveness to external sensory input? So.
Yeah, so that was initially my hypothesis, that there was sort of maybe a tuning into this internal sensory kind of field of information that either, maybe additively was overwhelming sort of in combination with external stimuli, or alternatively just was a way to sort of tune out of external stimuli that might be overwhelming on their own. And so some of what we're doing now is trying to get at that with self-report measures of things like intolerance of uncertainty, other anxiety traits, and that kind of thing. So and definitely lots of measures of repetitive behaviors as kind of appropriate. So so I certainly think that there are likely links here. I don't exactly know which direction they're in, but I think it's likely that there will be hopefully some connections we can draw.
Does anybody else have any other questions before we let Dr. Cascio go? All right.
I have a
Carissa,
question.
thanks so much. Oh, I'm sorry, Robin, go for it.
Okay. Can you hear me okay? I'm in the middle of nowhere, Kansas right now, so I don't know.
Yeah, I can hear you, but you do kind of sound like you're in the middle of nowhere.
Yeah. Okay. So, you know, variability in autism is a big thing, right? And I'm wondering, you kind of focused on this idea that, well, maybe there are no differences in certain aspects of interoception, or maybe there's a difference between children and adults. That may be measurement issues. Who knows? Have you given any consideration to other factors that may be contributing to heterogeneity, or have you seen the level of heterogeneity in your responses that is kind of classic of autism?
Yes, we definitely have seen a lot of heterogeneity in these tasks, but in fairness, I think we're probably a little bit ahead of the game compared to where we usually are, because typically developing people are also really heterogeneous in their ability to do the heartbeat counting task. So the relative difference is maybe not as striking as it can be for some other things. And I do think that we're addressing the individual differences issue is one that is always a challenge, but I think is helped by trying to get multiple measures, and that's really what we're really trying to do, particularly in this renewed R01, is to get multiple clinical report measures, multiple neural measures that include both MRI and EEG, and these behavioral tasks. And by sort of having a fuller picture of what each person looks like, I hope that we'll be better able to address how individual differences might play in.
Great, thank you. And it's excellent to hear updates on your work.
Thank you.
Stay safe, Robin. You sound like you are in the middle of nowhere or in a car or something. But anyway, Carissa, thanks so much for joining us. This is really cool work. I have to say, I mean, just thinking about it, there's just a number of ideas I'm sure you've been thinking about already. But the potential here for different forms of biofeedback types of therapy that might be able to address anxiety related or other specific issues. It's really fascinating work. So it's great to hear about it. I really wish you were here in Lawrence visiting. I'm sure Brian and other friends you have here wish the same. So hopefully we can get you to Lawrence when it's safe sometime in the near future. But great to see you. Thank you for joining us. And thank everyone for joining us. Hopefully we'll see many of you next week at our conference. Thanks again, Carissa.
Thank you all so much. I really enjoyed it. Good to see you guys. All right, you too.